All right. We need to keep going with viruses. So I think we left off here looking at what makes up a viral particle. And so you have some kind of capsid, and it can be pretty ornate or pretty straightforward. And then if you were to crack that open and peer inside of that, that's where you would find the nucleic acid and sometimes some special enzymes. And then, especially the viruses that attack animals, including us, they often have a membrane that they'll put around their capsid. And I think I mentioned on Friday, I always think about Harry Potter and his invisible cloak because it really does help keep the viral particle hidden from your immune system because essentially they wrap themselves in part of your body. And so your immune system doesn't really figure that out, at least not initially. And so, really good strategy from the viral perspective to stay hidden. So, when you look at all the different viruses that they've studied, you use this idea primarily, you hone in on their nucleic acid and you ask the question, what type? Is it DNA or RNA? We haven't found a virus that can do both. So, it's either or. And then, is it single or double stranded? And both DNA and RNA can be single or double stranded. So, it's kind of weird. So these are the different groups. I'm not asking you to memorize these. I'm just showing you that they have a classification system for viruses and to be familiar with like the basics of how they work, the classification based on nucleic acid. So here you're seeing a capsid with, you have many subunits of proteins that will make up the capsid. And, it's like a hollow core inside that you would find nucleic acid in there as well. Here they're showing the difference between enveloped versus naked. And they actually use that term, naked virus. That means no. So that's what you're seeing on the left. I don't care that you know the word capsid here. Okay, capsid, I want you to know capsid. And then you have subunits of protein that make that up, nucleic acid of some kind. And then sometimes they'll steal bits of the membrane and wrap that around themselves. And sometimes they'll put their own little proteins in there as well into the membrane. Different shapes. Some are pretty straightforward. The tobacco mosaic virus I mentioned, I think last time, was the most heavily studied virus before, I think, HIV. I think it was picked because, well, it was devastating to the tobacco plant and the tobacco industry. So there's probably a lot of money being thrown at it from that perspective, but it's safe. If you work with plant viruses, you don't risk getting infected. So that's a big win. You don't have to worry about it. If you get a little sloppy in the lab, you're not going to end up harming yourself. They have discovered some very big viruses in the permafrost that is melting. So giant viruses is a thing. The Mimi virus. It infects amoebas and it's large as some bacteria. And then it can become infected by viruses. So really, really wacky. So there's viruses that can infect viruses, which was kind of a shock. The Mimi virus is probably the most famous, but since then they have discovered some others as well. And so this has some implications for the evolution of life on planet Earth. They're asking what role viruses may have played. Did viruses come before cellular life or after? I'll talk a little bit more about that when we get more into the ecology. But basically, this is really, it kicked off with the idea of going into the permafrost. And we know that in the tundra areas, the permafrost is usually permanently frozen. And now, because of climate change, it's starting to thaw out. And one of the questions is there anything in the permafrost we need to be worried about in terms of microbes that we have not been exposed to for thousands upon thousands of years? Is this going to be an issue at all? And so one of the discoveries from the microbiology realm is, yes, there are these unusual entities The pithovirus has been discovered, mollyvirus. And so again, when they study these really large viruses, they have a lot of genes on their DNA. This one even has parts of a ribosome. So it really is kind of throwing a lot of questions at where do viruses fit in the evolutionary timeline? And do they, how did they fit in with cellular life? So, the common thought is that cellular life arose approximately 3.5 billion years ago. Some have pushed it to 3.8 billion. But now they're asking, where are the viruses in that timeline? So very interesting. Here's just trying to show you the number of genes on this one. And so it is a lot of microbiologists are starting to ask, could it be that it evolved off of a parasite, a parasitic microbe, and then evolved into a viral particle? So interesting questions going on right now in the world of virology on how they fit in to the evolutionary trajectory. So I'll talk a little bit more about that, but first let's just get into some other basics of viruses. When you go to study viruses in the lab, it's really challenging because we can't use ordinary microscopes for that. You always have to have a host to get it to make more of itself. So this idea working with bacteriophages has been very popular because, again, if you work with bacteriophages in the lab, you don't get sick from that if you make a mistake because they only infect bacteria. So that has been a long-standing model system because way easier to work with a phage and not have to carry huge health insurance. That's one thing, by the way, if you want to go work for the CDC, check out the health insurance package you're going to have to sign up for. That's what actually made me decide not to go work for the CDC. I was too cheap to pay those steep prices. You have to pay for some pretty healthy insurance if you want to work there. Anyways, animals, what do they do then? Well, again, they can work with animal viruses, but it's a lot more controlled and careful. You have to have a lot more, you have to go through a lot more hoop jumping in order to get certifications to do this. We're allowed to work with phages here, but you have to get special permission for any kind of animal viruses. If you're going to work with animal viruses, you tend to use tissue culture techniques where you work with the animal cell in a tissue culture process, where you can actually grow them up in like Petri plates. If they're animal cells, as long as you feed them the right nutrients, they can grow and divide and grow and divide and almost form like a lawn of growth in that Petri dish. It's usually a broth that you put into the dish. It's not like an algae surface. I worked with animal tissue culture when I was spent some time at MSU, and that was really interesting compared to how we work with it with plants or growing microbes in petri dishes. Very different. In plants, this is tough. Probably even tougher than working with animal cells because plants have a cell wall that's very strong, and viruses don't get in that way. Viruses don't, as far as we know anyways, there's yet to be a case where viruses can latch onto the surface of a plant cell and cause problems that way. They have to get in like through a wound or through some kind of opening, and an intact plant cell won't allow for that. So what you have to do is you take the plant cell and you have to degrade off, very carefully, you have to degrade off the cell wall and leave just the cell membrane and the internal contents hopefully nicely intact beneath that. So that's called a protoplast. So if you hear anyone say the word protoplast, they're talking about plant cells that have the cell wall stripped off of it. And so that's a little extra technique that you have to do. So they grow the viral particles, so to speak. They allow it for them to replicate, might be a better way to say it, in the host cell, whether it's animal, plants, or bacteria, for example. And they can track the replication cycle. And it does somewhat look like a growth curve that we've already talked about for bacteria, but they talk about it a little differently. They use other terms like an eclipse phase is kind of like the lag phase, or yeah, the lag phase, sorry. And then you see this rise period, and that's kind of analogous to the exponential phase that we talked about. And then this leveling off is basically everything's done in terms of the cells have been lysed and the phages have burst. And so you can do some simple calculations to figure out how many viral particles per cell and how many are in that population that you started with, for example. And so you can use this information similar to what we do with growing bacteria in a batch culture. And so you can get some good data from this, and that's how they figure out like that, different viruses will have a different burst size essentially, and it just depends on the virus and the host cell what that number is going to be. So you can characterize that. And then the red graph, yeah, they're doing both intracellular and extracellular. So you can work with these systems like this in a batch culture, and you can try to quantify. But it's an estimate for sure. It's not super accurate, but it gets you some good data. They often will say, we'll talk about virus infectious unit. And so we usually say colony forming units when we talk about bacteria populations. They talk about viral infectious units. And if you want to track them more across a plate, like, with what we've been doing in lab way back when we did the spread plate exercise, where we gave you a test tube with some microbes, some bacteria in it, and you diluted it, and then you aliquoted it out, spread it around, and then you counted colonies. Well, in the viral realm, what you do is you run a plaque assay. That's the viral version of a spread plate assay. So what you do is, so if you have a bacteriophage, you can grow like E. coli, for example, and you will spike a dose of E. coli with the virus, and then you can spread that out onto a plate and incubate and look for plaques. So basically what you're doing, and we used to do this in the lab. It's not too hard to take E. coli is usually the bacteria, and you infect it with some phages, and then mix them together, and then you pour it into liquid agar that's been cooled down, and then you pour that all into a plate and let it solidify. And as you're incubating that plate, the virus is busy infecting and killing E. coli. And any place that they successfully got into E. coli and did their replication cycle, they'll leave a little tiny clear zone in that soft agar. And so that's, instead of a colony, you're counting a clear zone which is essentially a kill zone that the virus, ended up killing off E. coli or whichever bacteria. So you refer to those kill zones as a plaque. You can do this we call it soft agar. You're growing E. coli in agar where the amount of agar has been reduced quite a bit, similar to the plates where we do test for motility, and we use motility agar. It's similar to that idea. That allows for nice dispersion of E. coli and the virus, the phage. So this is the lab. You have bacteria, and then you put it into some soft agar, mix it all together, pour it out onto the plate. You usually have what's called a base plate, which just means you have already, that agar plate has a base layer of agar, and then you mix the soft agar, the phage, and coli together, get that all nicely up for and incubate. And then what ends up happening is the phage will go through the replication cycle, and as they are done replicating in each cell, they'll break open, they'll lyse open the bacteria, and they leave a kill zone. This is another image of that idea. So this is what you would see when you take your plates out of the incubator. And sometimes you have to dilute your phage sample because it'll be too concentrated. You'll get like total kill across the whole plate, and then you can't make any counts. So again, you're trying to get that 30 to 300 range of plots in this case where you count, and then you can use that number to figure out what your population size is in the original tube that was provided to you. And so this is a way for epidemiologists to figure out, you know, how sick a patient may be. They can quantify that and get a little bit better feel. So with bacteria, it's pretty straightforward because very easy to visually see plaques, because all the rest of that problem is E. coli that was not infected. And so it provides a nice cloudy background and then that very definite clear zone. It's a little tougher. Oh, and then there's some kits you can buy where you can stain as well. So it's easier to pick out that way. If you don't want to do the more typical way, you can get the fancier kits. So there's that option as well. I like this picture because it's pointing out the idea that not all plaques are identical. And it seems to be pretty unique. Each virus tends to have a pretty unique looking plaque. So that's another way to try to figure out what this viral particle might be, at least to some extent. You could get a little better feel for that. So, I mean, you could easily see that these plaques look different from each other. And so that information could be very helpful as well. Animal viruses, at least when I was looking at plates myself, I don't have a ton of experience with animal viruses, but it's just a little harder for me to interpret the data. I think once you train your eye, it's a little bit more straightforward, but in this progression, you can definitely see a more congruent lawn of cells in the 0 hour, and then you start to see the disruption happening over time. So with animal cells, it's a little different look to it, but you need to be a little bit more gentle with this because usually the cells are growing on top of a liquid rather than an agar base. So it's a little trickier to work with. And then I work with Streptomyces bacteria that form, you know, colonies on plates like most bacteria do, but they have that powdery look to their colony. And I have isolated hundreds of Streptomyces bacteria over the years from soil samples primarily or plant roots. And I would often see this look to the colony where, you know, normally it's this very fine powdery look after several days of incubation. But I think you can see, especially with this one, you can see all these tiny popcorn green, but you can see that. I used to see that all the time and didn't really think much of it. But it turns out when they started looking at those are actually phages that are infecting those colonies. And that kind of was a light bulb moment for me because when I was in graduate school, one of my Streptomyces bacteria stopped producing antibiotic when I desperately needed it to produce antibiotic. So I went into a huge panic mode because I needed to keep doing that to finish my research. And I'm starting to wonder, you know, when I learned about this, that maybe the phages were somehow involved in messing that up. So luckily I had a frozen culture that still could produce antibiotics. But so look for that in any of the colonies that you may be growing, you see in lab through the rest of the semester. You just never know. They may be lurking. Phages are everywhere. So again, we've already talked about this idea that if you're trying to quantify microbes using, counting colonies, we tend to underestimate. And if you really want to get accurate, you buy an electron microscope and look at the viral particles that way, but that's super expensive. So that's pretty limiting. If you do the normal plating that we were just looking at, we know that it's not a high success rate. So we know that the viruses aren't always going to behave optimally because you're putting them into a very unusual artificial situation. So we think there's a pretty low success rate of infection. And so we just kind of recognize that, but it's still useful data to get. And it's more just, we don't know quite how to work with viruses well enough to get to optimize them. So we just assume that we're underestimating pretty much all the time. And that's a pretty safe bet. And so sometimes when they spit data back out, they'll talk about plaque forming units, and it's the same idea as colony forming units. They're counting plaques instead of colonies. And then they'll talk about titers. You might hear that phrase. That's really just the concentration. I don't know why they have to use the word titer per se. It must be a medical thing. But it's really, if they say there's so many PFUs per titer, blah, 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 that's just their concentration. That's a concentration unit. So don't get too lost with that one. Okay, so viruses are challenging to work with in the lab. Either you need a really expensive electron microscope, or you have to know enough about their host, and then you have to know how to grow the host cells, plus make the virus happy enough to infect it. So it's challenging. It's a lot of work. So here they're trying to show that you have this idea of a lysis event occurring after the end of the replication process. That's what's being shown on the top there. And then the other idea too that we need to think about, and we've already talked about it actually, is that we know, especially when phages are infecting bacteria, they certainly can paste their chromosome, their genes into the host DNA. And so the virus or the phage can kind of be hidden in the cell that way, because when their DNA is pasted in there, they're not in an active infection cycle. They're just hanging. But now if that cell decides to go ahead and replicate itself, all progeny cell will have that viral DNA. And so it's kind of a sneaky way to spread by just hanging there, getting the DNA pasted into the host DNA. And so we've talked about this idea already. Let's go through the step-by-step here. So with the viral replication, we'd like you to know the 5 steps. This is outlining the 5 steps, very basic. We're not going to get into huge detail on this, but basically, in order for a virus to do what it needs to do, it has to attach to the host cell, and the right host cell, can't just be any, right? So there's usually very specific surface receptor proteins that the virus has evolved to interact with and dock into. And so that's why when you get a cold, the cold virus is in your nose throat area. So those receptors of those cells, the cold virus has adapted to. But if it accidentally got into your kidney, for example, it wouldn't cause a problem in your kidney because the surface receptors are different. So that attachment idea, they have to attach. First, they got to do that. And so a lot of effort is looking at how do we stop viruses from attaching in the first place. If you can stop that, you win. They'll never be able to cause problems. And so that's a huge part of trying to fight on viruses. You can figure out a way to do that. Well, let's say they do attach. Sometimes they call it absorption. The next step is penetration or injection, and that can vary. And we'll look at a few examples. How they do that can vary, but basically they have to at least get their nucleic acid in. At the very least, they got to do that. And then what happens if they can do that successfully, then they go through synthesis. You don't say biosynthesis because most people don't think viruses are alive. So you drop the bio term, you just say synthesis. And that's just making more bits and pieces of the virus particle. And they're trying to show in this drawing that it's all just bits and pieces. And then the fourth step is maturation or assembly. That's where you take all the bits and pieces and put it into a coherent whole viral particle. And that's what they're trying to show in that drawing. And then the last part is release or lysis, where the viral particles get out. And very often the host cell lyses and dies. So please know the 5 steps, the basics. So all of this can be tracked. You can, and they know the data on lots of these different viruses. In general, with all the bacterial viruses that have been studied, the range is roughly 20 to 60 minutes from start to finish. With animals, it's complicated because the animal cell type is more complicated. It just takes longer to do everything in a more complicated cell. And so that can go from 8 to 40 hours from start to finish. Here, they're just trying to map out what's happening in relative to the so-called growth curve, so to speak. But I'm not going to get into that. But here you can see, it's until you get that lysis going on, viruses are very good at hiding away from the immune system, at least initially. And so that idea that if you can somehow stop the attachment phase, that would be like the gold standard drug therapy. You could do that. And so I guess one of the upsides is the specificity. Most viral particles are super specific on what they can interact with on the host surface. And so, for a complex organism like us, we have different tissue types and all with their different receptors. And so all of that plays into, so if a virus gets into your body, if it got into the wrong part of your body, you'd never know it because it wouldn't cause you any trouble because it wouldn't successfully attach. And so it really does have to find the right cell type in order to cause problems with you. And so that high specificity that their capsid, or if they have certain appendages, tail fibers or something like that, where they can use those to dock into different, it's usually a protein receptor on the host cell. So if they can do that, then they've gained a foothold and they can start getting into the next phase, which is the penetration or injection. And depending on the virus, it's going to either be just the nucleic acid or maybe the whole viral particle. It just depends. And I'll give you some examples. And then don't forget, if the host cell has a cell wall, that can be very problematic for the virus. So, and like I said, plants anyways, we don't really see viruses doing much of anything. The cell wall pretty much precludes. They have to get in. Usually viruses get in to a plant with the help of an insect, or if there's damage, then they can get in. But if they are presented with healthy plant cells, they don't seem to be able to do it. So here you can see a really nice image of, I believe this is E. coli, on the right there, and you can see the phages on the surface of the cell. So this is that T4 phage that looks very much like it came from a science fiction. I swear science fiction movie directors, they probably have a microbiologist on staff to come up with ideas of some of the sci-fi movies I've seen, because I'll watch some, especially some of the sci-fi movies that are not super popular. They kind of bombed at the theater, but so many of some of the sci-fi I've seen, it's like, oh, that looks like microbiology stuff that they're pulling their ideas from. So here you can see that there's a few of the phages on the surface that are docking into the receptors. So these tail pins, they call them, and tail fibers are often what's used in the more ornate phages. So here they're trying to show the interaction where you have, high specificity where those tail fibers and tail pins will interact with very specific molecules. And if they can't find those molecules on the surface, they're not going to be able to do the next step. But this one did. And you can see in this particular example, there's a lot to it. And we're not going to get into detail. See how and they kind of use the analogy of like a syringe or a hypodermic needle, where you have this injection of the DNA connected to the compression here, that part of the phase where that helps push the nucleic acid through. So pretty complicated, pretty amazing how successful that phage can be. And so all the rest of it stays outside. Here's another image showing how it's falling off. So once it injects I don't know if you can see with the light in here, but you can see there's a little bit of bluish, a strand of blue, that's the nucleic acid that's getting injected in. And then you can see the ones that had already done that, the rest of the phage just falls off. Seems like kind of a waste, but it works apparently for them. So very kind of complicated attachment phase, and then the bulk of the particle is just done. It doesn't make it into the cell. They just inject their nucleic acid. Here on the right, figure B, you're seeing a little different situation where you have the docking going on, and they're trying to show that the whole thing got in, not just the nucleic acid. So the whole viral particle was taken in. And very often what that does is it's through the endocytosis process that allows for that, where the viral particles, they have to attach first. And then that triggers the cell. If there's the correct docking, if they can correctly dock into a surface receptor that's kind of tricking the cell into like, hey, I'm innocent. I docked into you just perfectly, so this is meant to be. Let me in. And then the endocytosis process kicks in and allows for that kind of evaluation and the pitching off. You have a vesicle. So the lower image is electron microscope process. So again, that attachment can be, it's like the lock in a key idea. Once you attach, that can kick things into motion that the host doesn't want, but it's very successful for the viral particle. Here again, they're just trying to show the surface receptors, very specific surface receptors for each viral particle. So the rhinovirus is a common cold, HIV on the right. Okay, so now we are very complicated organisms. So luckily we have a very sophisticated immune system, of course, that can fight back. And, Dr. Sharp teaches a whole class in immunology and then also virology. So I'm just giving you an overview of what you will find in those classes. So with bacteria, initially they thought bacteria had no immune system. Now we know that's not true. But generally they'll say it's not as sophisticated as a multicellular organism, but they do have those restriction enzymes I already talked to you about a while ago when we were looking at how phages can get in earlier with gene transfer ideas. And so restriction enzymes, we actually have bacteria to thank for all the restriction enzymes we can purchase and use and research. And so these are specific only for the viruses that have double-stranded DNA. So out of luck if it's not that they're specific for double-stranded DNA. And then viruses have learned to fight back because they can kind of hide their DNA from the restriction enzymes. They can often do that through putting sugar units on the DNA, and that prevents the endonucleases from cutting where they're supposed to cut. They can methylate their DNA, and so viruses are very quick to evolve strategies to try to one-up the situation and gain benefit over the whatever immune system process is getting thrown at them. So very, very quick to evolve and adapt. They're amazing that way. And then there's some viruses that even carry proteins with them that basically is stopping the restriction enzymes from doing their job. So viruses are even more quick to adapt than bacteria. And so they talk about this arms race that's continually going on between the host and the viral particle. Here's some images trying to show some of the modifications. So glucosylation there in the lower image on the nucleotide base. Here they're showing the methylation going on. So if you can do that, use effectively now those restriction enzymes can't get in there and do what they're supposed to do. So that's, again, this arms race idea. Then you can see that the host cell can modify, can adapt to this issue where the virus has, gained an upper hand. Well, then the host cell, in this case, bacteria, they can start adapting and evolving. And so they've seen where some bacteria will gain genetic resistance that allows them to modify their surface receptors so the phage can't even attach, which is awesome. If they can't attach, they can't cause problems. And then CRISPR, we think we have bacteria to thank for that. And so CRISPR is part of their immune system. That's what it's really for. We take advantage of it and are using it in all kinds of applications and amazing applications going on right now. Seems like every day there's something new going on with CRISPR technology. But basically that stands for clustered, regularly interspaced, short palindromic repeats. Good gravy. So they put the acronym CRISPR, and it really is an immune system for bacteria. It's their version of an immune system, and they say it's really kind of analogous to the human adaptive immune system. It's much simpler, but it can be pretty useful for the bacteria. So this idea of the virus can evolve and adapt and overcome, but then the bacteria can also do that. And so there's this constant back and forth between, they've studied a lot, bacteria and the phages that interact with them. So this is the idea of CRISPR. I'm not going to get into it, but I've had students ask about it in the past, like in there, but basically through action, it's a way for the bacteria to be able to pick up this foreign DNA that's coming into them, and they can recognize it and then deal with it and chop it up. All right, and so and now we've taken that and have modified it, used it in molecular biology applications. So if you're curious, I'm not really going to get into asking you about CRISPR other than just knowing it's a bacterial immune system process. Do you learn about CRISPR in 218? I would think by now they at least talk a little bit about it. It's something that's going to impact your life one way or another, or someone that you love one way or another. It's an amazing technology. Okay, so now, if the viral particle can attach and penetrate all or part of itself into the host, then the next step is synthesis, where it needs to make more of itself. So this is an overview of how the different groups of viral particles do that. Okay, so let's just step back a second. When we think about replication of our own cells, you've been taught the idea that it's DNA to RNA to protein. You have to have the genes on the DNA transcribed into RNA and then translated into proteins. Okay, well, the virus has to do that too, because the virus is made of nucleic acid, the virus is made of protein. So it's got to make its own proteins, including its own enzymes. It's got to make the proteins that make the capsid. It's got to make the proteins that make any of the tail fibers or whatever appendages they might have. Then it's got to make its own nucleic acid, whether it's DNA or RNA. So it's got to build all of that using the host machinery. And so eukaryotic cells, bacterial cells, it's DNA to RNA to protein. That's the process. Okay? So we have to think about if the virus particle is a DNA virus, it's pretty straightforward. But if they're RNA viruses, then it gets a little wiki on how they do that. So I'm going to kind of break this down a little bit. Let's go to this. So this is what I was just talking about. If they're in the replication phase, they have to make more viral particles. So it's nucleic acids, they have to make more viral nucleic acids, more viral proteins, any viral enzymes. So if it's a double-stranded DNA virus, it's usually pretty straightforward because the host is that way, right? Well, not. The thing about us, we're double-stranded DNA. So going from double-stranded DNA, messenger RNA, and protein, that's what we do. And so the virus, if it's a double-stranded viral particle, not too difficult. It has the proper machinery, it's pretty straightforward, where it can just take the transcription and translation machinery hostage and say, make my nucleic acid, make my protein, make my enzyme. And so that's pretty straightforward. Okay. So double-stranded viral particles, pretty straightforward. If, on the other hand, you're a single-stranded DNA virus, the first thing you have to do once you get into the host is make a complementary copy. Again, you'll use the host machinery process to do that. So the replication process to replicate DNA, you'll grab onto that and say, well, I'm only single-stranded, make a complementary copy so I get into the double-stranded state, and then I can take advantage of the host messenger RNA process, the transcription and translation process. So basically, they have to kind of get into the form that the host machinery can easily work with. So in this case, with animal cells, you have to get into the double-stranded DNA. Okay, then it gets a little wacky with RNA. And okay, the other thing too, do I have that? In the notes section of this slide, it's I put a little additional note. So we talk about plus and minus RNA with viruses. So by convention, if it's called the plus RNA, that means you can use it directly as messenger RNA. You can translate off of that and get your proteins made. Okay, so that's our jargon for if we say it's a plus strand, then what we're saying, you can take that plus RNA and you can translate it into protein. Okay. And then it says in the notes, where is that? For example, a virus that has a single-stranded RNA genome with the same orientation as the messenger RNA, then it's a positive strand RNA virus. A virus whose single-stranded RNA genome is complementary to its messenger RNA is called a negative strand virus. All right, so we're going to look at some examples of that as well. So if it's a positive sense RNA, I mean, usually what we're saying there is that it has on it the necessary information it encodes for viral RNA-dependent RNA polymerase. So what that means is it can make an RNA polymerase off of a stretch of the RNA and then use that RNA polymerase to make more RNA. All right, and so it can do that. And so that RNA polymerase is very important. Once it gets that made, then what it does is it allows that RNA polymerase can read the plus strand, and it's going to make a complementary minus strand of RNA off of that plus strand. So if it's complementary to the plus, then we call it the minus. All right, so plus and minus are always complementary to each other. So what happens is first it will make this RNA polymerase, and then it uses that RNA polymerase to walk down the plus plus strand essentially, and make a complementary copy off of that. And then we call that copy one. And that's necessary because the virus has to get that minus strand made. And then what it does is it'll use that minus strand to pump out a bunch of plus strands off of it. It uses it like a little factory. It says, okay, now I got my minus strand template. I can use that to make complementary plus nucleic acid. And so it'll do that over and over and over to get a bunch of its positive center. A little bit more complicated than if it's a DNA virus. Okay. All right, let's stop there. That's kind of a gnarly thought. We'll get into that again on Wednesday and we'll go over a couple of other examples.